right? Moving forward. We're good. We are live. All right. Entamoeba. All right, this is where we left off. Causative agent of amoebic dysentery. Right, so we went over the life cycle. I think it's kind of interesting in terms of the cysts. You know, with Giardia, you could have any age of cysts come out, and in theory, they could all be infective, immediately infective, or they can all be infective if they develop properly. With Entamoeba, it has to get to that metacyst stage because that's the only stage that can actually survive outside the host. All right, so we had our transition to the precyst and the young cyst and regular cyst, and then the metacyst. Metacyst leaves, you ingest it, and then you have it excists, and then it divides. And it's those stages, those metacystic stages, that have to find their final site of infection, which is in the crypts of the large intestine. So then once they get there, they can replicate. So that means, you know, our life cycle is fecal oral, but the infection success, whether or not we get infected, is going to be dependent on the dose that we receive. So if you just get a single metacyst, you're unlikely to ever, it's going to be less likely that you get it. But if you end up ingesting 100 or more, chances are you will get it. Something similar happens with Giardia, but in that case, it just kind of depends on the strain of Giardia and what your immune status is. So we could drink the same water. We can go out on a hike and we could grab a cup of water from the river, all right? And I can get Giardia, but you might not. And that's kind of that big question. Why does one person get it and suffer while everyone else does? Because that one person stepped on that one endangered plant. It's coming back for you. Oh, hold on. Forgot. All right, so before we move on, that's where we left off. Parasite in the news. It's popped up. This was interesting. I don't know if you saw it. Corpse flower. So I know I had mentioned it, that there are parasitic plants, right? What parasitic plant do we have here on campus? Readily visible. Mistletoe.
Oh, that's her.
Mistletoe. Another Christmas plant, right? Yeah, this is the corpse flower. The flowers are, I think, one of the species is the largest flower known. All right? And why it made the news is because they've sequenced DNA and then also had another group sequence like similar species of DNA, because when that one group was trying to sequence chloroplast DNA and all that stuff, it's missing all of these photosynthetic synthesis genes. It's missing all of them. And if you think about it, evolutionarily, it kind of makes sense, right? So these are parasitic. They form a really small line of cells inside the tissue of the plants. They're so small that most people will overlook them. You have to really be looking for these cells in order to identify them. Then eventually, all of a sudden, you have one of these flowers pop up, and they're putrid smell. That's why they're called the corpse flower. They attract carrion-feeding beetles, but they're missing a whole lot of genes that all of the other plants have. So, people are like, well, why would you lose it? Well, I kind of have made it a point in the ecology class to say, if you don't need those genes, why keep them? Just like here in parasitology, if you don't need a mitochondria, why keep it? It's just extra upkeep. So evolutionarily, this plant probably became effective as a parasite, to to where not only was it becoming entirely internal, with the exception of the flower, but then, since it's not photosynthesizing, it just drops those genes, drops those functional genes altogether. So yeah, this was in the yeah, and they had other ones that also looked, so you can see that which types of genes popped up. But this is the corpse flower, so it's parasitic. Check it out if you're curious. That was in the news.
How big is the flower?
Up to like 1 meter wide. Here it is. It's here. Yeah, so foreign fibers can be more than 10 meters long. That's in the tissues of the plants. It's a single line of cells. I think this one talked about the flowers, how big the flowers are. They can be big. I remember the size because they talked about the size of the flower weighing like up to 10 kilograms. And the author's like, that's like the size of a toddler. I just chuckled. But that's cool. Cool. I wonder. I did a search for news. It's probably on Wikipedia or something like that. Yeah, corpse flower, corpse plant, different ones. So they're big. They're big. But you can check it out. Here it is. Measured 3.1 meters tall. It's 2.94 meters. Pretty cool. I think it would be cool if we had one here in the greenhouse.
Were we trying to get one?
What's that?
Were we trying to get one in the greenhouse?
I don't know. I don't know. Just to have that. Then we could put out a news release. ASU Biology has a dead body on campus. You know, we'll send it out as like corpse. We have corpse, but not say it's a flower. So then, but St. Angela Alive would probably run with that pretty good, right? All right, so we talked about the life cycle. Let's go over the pathology. So amoebic dysentery. I'm sure you've heard of dysentery before. Might not really know what all of it is about. Well, Entamoeba causes it. So the pathology, how severe, how long you have it, is going to be strain dependent. It's going to be host dependent. Some of our strains are super severe, or some of our strains are more severe than others. So some of them are going to be minor, others are just like nasty. And again, if we get it at the same time, maybe it's severe for me, but not so severe for you. All right. The pathology is all tied to the parasite. All right. And in this case, I think I probably would call it damage. You know, mechanical damage. It's chemical damage is what it is, but it's damage. So the parasite is feeding on, gut contents. It's feeding on host mucus. It's going to release enzymes to break down some cells, break down whatever's out in its environment so that it can absorb the nutrients. Some of that will cause tissue to lyse, the actual host epithelial cells. All right. When you do this, now you have the possibility of forming ulcers or lesions in the gut that now exposes you to secondary infections. All right. These typically form initially in the coelom, in the cecum, but they can also be in the appendix and then also in the upper colon. As parasite population increases in that region, you're going to have it spread, which causes even more destruction of the mucosal lining. And normally it's going to be a spreading laterally along the length of the colon, so wherever it initially started to form. So I do have some images here. So pre-cyst. This could be a pre-cyst or a young cyst. It's hard to tell if there's a cyst wall on there or not. But you can see the nucleus and you can see this is probably a cyst, a young cyst. 2 nuclei, I think, are developed along with their chromatoidal bar. So you ready? All right, so the lesions and the feeding, hydrolysis, leads to symptoms of amoebic dysentery, which is explosive diarrhea, bouts of abdominal pain, dehydration, loss of blood, potential death could result from gut perforation, or death could occur from cardiac failure and exhaustion. Just your body's tired of fighting it, and everything just kind of shuts down. All right, so you get all these symptoms. I mean, they are somewhat similar, a little bit similar to Giardia. I think with Giardia, you're less likely to see some of the bloody diarrhea, but you definitely have the diarrhea, dehydration. And with Giardia, you don't get gut perforation. Typically don't get gut perforation. Now, when you have these bouts of diarrhea, you're going to find the trophozoites, the precysts, and the cysts in the stools. All right. You're going to find it even if it's not really full-fledged diarrhea. If you only have partially formed stools, you can find the trophozoites, precysts, and cysts. The only time you're going to find those metacysts is if you're not really exhibiting these symptoms. Because if you're not exhibiting these symptoms, then you're probably developing fully formed stools. And that's what you need, because you needed the signal to cause cysts to start forming, and it needed to happen early enough so that the metacyst could form before it leaves the host. All right. So this is just kind of like another reason why you should wash your hands. Have people, if you're going to prepare food, wash your hands ahead of time. You never know. And I think a lot of people say, oh, I'm not feeling good, so I need to make sure my hands. Well, you're not going to get amoebic dysentery. The metacysts aren't there. It's the normal feeling, normal appearing people that might be spreading it. This is a metacyst. There's actually 4 nuclei. This is what was on one of our slides. I took that picture. No chromatoidal bar, and I do have, you can see 2 nuclei. There's 2 more nuclei in there. You can see it if we find focus through the cyst. So these start to go out of focus, and then you find the second set of nuclei, and I can't remember if it's like back here or back here, but you can see all 4 nuclei. Ready? All right, gut perforation is probably the worst case scenario, because once you get the gut perforation, now the Entamoeba can spread. And when it spreads, it can form lesions in whatever sites they colonize. And so just these ectopic lesions. This gives us a little bit more specific terms. So amoebiasis or amoebic dysentery would be Entamoeba. But if we have hepatic amoebiasis, it's also, it's basically amoeboid infection, but this is on the liver. So the amoeba spread, trophozoite spread to the liver. It's still going to hydrolyze those cells. It's still feeding, absorbing stuff. It causes lesions to form. Pulmonary amoebiasis, as you hopefully can know, is when it forms in the liver. Now, usually the hepatic amoebiasis would develop first, because it's the amoeba are in the colon, in the large intestine, and then when they penetrate out, they're normally going to get to the liver first. They cause that, and then from the liver, you can get some penetration through the diaphragm that ultimately allows the parasite to get to the lungs. It's notable that in these situations, precysts can't form. And if precysts can't form, then you can't have any of those later cyst stages. Why is that? What's the signal for cyst formation?
No water.
Yep, water uptake. So as it encounters its dehydrating environment, that's when it's going to start forming our cysts. And that's not going to happen in the liver. It's not going to happen in the lung or other places. So I do have image from a textbook. This was actually a fairly big image. It took up like more than half the page. Shrunk down a little bit, but I copied and pasted it into our handout. So it kind of shows you the different lesions, destruction, and so forth. Questions? ready to move on? All right. That wraps up our amoeba. I think we have a presentation on Naegleria fowleri. That's also an amoeba, but that'll be in one of our species presentations. So, talked about our flagellates. We talked about our amoeba. And now we're moving to the last part. The alveolates, or I should say, the phylum Apicomplexa. Phylum Apicomplexa. All right, the Alveolata. Again, kind of moving into our modern age systematics. Members of this group possess cortical alveoli, which are sacs. Hence the name, Alveolata. You have these sacs. Also, you have these flattened vesicles that form a continuous layer just under the cell membrane. It's thought that this continuous layer of flattened vesicle helps to support and give the cell membrane structure. So it's not going to be like an amoeboid shape. Right? We're actually forming a shape in what I would call kind of like a banana-shaped structure. Banana-shaped organism. So we've got these alveoli. We have micropores. We have mitochondria. But the mitochondria in this case have tubular cristae, which we've seen that before, right? We've seen tubular cristae before. This group is going to include our dinoflagellates, the ciliates, and Apicomplexa. So I think some of you, looking through the snails, have seen some of the ciliates. We didn't really look at any dinoflagellates. What are dinoflagellates? Don't tell me members of the Alveolata. They are. They have a hard shell, hard test on them. Yeah, with like a flagella, maybe some cilia out, and so forth. And then our Apicomplexans. So we're going to focus on the Apicomplexans because that's dinoflagellates are free living. Ciliates. You've got free living, you have commensals, and you have some parasitic forms. We're not going to talk about ciliates. We're going to focus on the Apicomplexans. Why the Apicomplexans? Because these have some of the most important human parasites around. Yeah, I know we've talked about a lot of important parasites. This is the group that you're going to see. You've heard it. You've probably heard them. It can cause death. Could cause death pretty rapidly. Could cause other symptoms. So we're going to spend some time on the Apicomplexans. This phylum contains a large number of species, more than 5,000. More than 5,000, there's possibly even more than that. It's just a lot of, you have to be looking for them to find them. And sometimes morphology isn't extremely distinct. So I think there's a lot of species that are undescribed, or at least have been classified as some other species. Some members are very important to human health. Example, malaria. That's an Apicomplexa. Toxoplasma gondii. We heard it talk in our seminar class. That's an Apicomplexa. Right. All members of this group, they're called Apicomplexa because they possess an apical complex. Apicomplexa, apical complex. I have a diagram here that kind of shows the structure of a typical Apicomplexa. All right, so what does the apical complex consist of? So our apical complex is at the anterior end. Its function is to help enter our host cells, because these are going to be intracellular parasites. First off, that complex consists of a polar ring. So you don't have it like a ring-like structure, but these things connect, both sides connect to form a ring around right at the very top. Now, it's not just one ring, it could be multiple rings. These are anchor microtubules. They're going to support all of the rest of that apical complex. All right, it's going to help hold everything in place. All right, inside that ring, that polar ring, we have what are called conoids. Conoids, so they're located up here at the top, have a number of coiled microtubules that exist as fibrillar cones inside that polar ring. It's thought that these aid in penetration. In some way, they aid in penetration. Ready? Ready? All right, next up for our apical complex are these glandular structures. These are called rock trees. These are tubular or saccular organelles. Their function is in entry, but more specifically, proteolytic entry. So they're going to secrete their contents to help break through the cell membrane of the host to get inside of them. And our last structure of the apical complex are these micronemes. They are kind of scattered here. These are elongated tubular organelles. They're also possibly used in penetration. Once our parasite stage enters the cell, this apical structure disappears. It's no longer needed. That's why we know the function of some of these things, but the others it said may aid. And if it doesn't aid in penetration, why would you have it, and then why would you lose it? It's kind of the idea. Ready? No? Sorry. I've got an image on the next one. Yeah, an image and diagram. All right. Trickery. Not all of our species have all 4 structures of the apical complex. But this would be one. This would be a diagram of Toxoplasma gondii with things labeled. So you can see the apical rings. All right. Apical rings. You've got your microtubules. All right. And then you've got these subpellicular microtubules that help form the structure, give it shape, give it rigidity. Not on here are the rock trees or the micronemes, but I think it kind of demonstrates what those rings look like. Again, kind of helps support. And that's been posted. All right, so what else is in these apicomplexans? We've got micropores. Here's the micropore here. We're going to find those in the sporozoite, merozoite, and the trophozoite stages. Its function is similar to that of a cytostome. So what's the function of a cytostome? Cyto means what? What stone? Yeah, so what do you think it's going to do? Endocytosis? Or some modification of it. Right, endocytosis is just our generic name. All right, these parasites in the apicomplexan reproduce sexually and asexually. But I don't want you to think that we're going to have our female system and our male system. With our sexual reproduction, just again, generically, we're going to have 2 gametes that fuse to form a zygote. All right? And that means then you're going to have meiosis to reduce the number of chromosomes, to reduce our genetic code back down to the haploid number. Now, usually, you're going to be haploid, or these parasites are going to be haploid for most of their life cycle, for nearly all of its life cycle. So when the gametes fuse, form that zygote, normally one of the first steps is to do zygotic reduction, zygotic meiosis, right, to get back to the haploid stage. So that's our sexual reproduction. And then asexual reproduction is going to be usually our multiple fission events. Many of our species have what are called oocysts. These have also been called spores. And these would have a cell wall. Many species have cell walls. And that means then that if we're going to produce these oocysts or these spores, then it's probably the transmission stage because that cell wall will confer protection in the environment. Plasmodium has basically an oocyst that forms, but it doesn't have the cell wall because Plasmodium is not being transmitted through the environment. All right, we ready? All right, so there's two classes of Apicomplexa. All right, Conoidacida and the Aconoidacida. Note the A. All right, so difference is whether or not conoids are present. So in the Conoidacida, yeah, conoids are present. In the Aconoidacida, conoids are not present. They're absent. Generally absent. Generally absent. All right, so in this classes, we have a couple groups. So we have the Gregorins, which is what we're going to be talking about next. That's in subclass Gregorina, Gregorinacea. The Gregorins. All right, second subclass are the coccidian parasites. All right, so those are in the conoids. We're going to talk about those. Coccidian parasites include Eimeria and Toxoplasma. We'll talk about those. The Aconoidacida includes two groups. The two orders that we're going to encounter. Plasmodium, that's our malaria. That's a Hemosporida. That's some blood parasites. And then we also have the Pyroplasmorida. That includes Babesia and Filaria. These are possible presentations. So Babesia was presented. It's been posted up on Blackboard, or at least the link to the YouTube video. So check it out. We're not going to talk about these at all, but I add them here because I know that they're an option for our species presentations. Ready? All right, so we're going to start with the Gregorins. So this is class Conoidacida, order Eugregorina. These Gregorin parasites are going to parasitize invertebrates, primarily annelids and arthropods. And this is another group where it seems like whenever an individual has looked through an insect species, it's almost like they are describing a new species of Gregorin parasites. And something we could easily do. Go out and catch those grasshoppers that we did in ecology, bring them back and do some gut mounts and look for these Gregorin parasites, gut smears. Something we could do. Our transmission is going to require consumption of oocysts or spores. All right, so we're going to have exit out of the host, and then the host is going to pick it up, which means that we are dealing with a direct life cycle. Merogeny is absent in this group. All right, so merogeny is the production of merozoites. Okay. We haven't encountered that yet. We haven't encountered multiple fission. We are. We're going to see it now. Now, in this group, we have two orders. There are two suborders. Suborder Aseptoturina and suborder Septoturina. So again, note the A. So the A means lacking, right? Without. So Septoturina is a situation where our gametes are in two parts with a septum between the two parts. So our gamete consists of 2 parts with the septum between them. Aseptoturana doesn't have that septum, so that means our gamete is not in 2 parts. It's in a single body. All right. The 2 species that we're going to talk about are Monocystis lumbrici and Gregorino cuneata, or Gregorin sp. But we're going to do the cuneata because it goes into flower beetles, mealworms. So I'm going to try to pick up some mealworms too. Monocystis lumbrici is in our earthworms, night crawlers. Everyone that we've looked at has had them. Every year. Everyone. All right. Ready? Not sure when we do our life cycle. Ready? Almost. Yeah. All right. Monocystis lumbrici. You're going to see that we're basically just describing life cycles. Going to look at some of these stages, introduce terms, different terminology, because the pathology is, if it doesn't affect humans, it's like, who cares? I think some people care, but not everyone. Not the people who control the first. So, enlarge this, and let's do our life cycle. Let's hope we can. Hoping I can get in this little space. All right. We'll check it. Monocystis lumbrici. Almost. All right. So what we're going to start with are the mature spores. The mature spores, these are also our oocysts. Our mature spores are considered mature because each one has 8 sporozoites. This mature spore gets consumed. Consumed by our host, which will be an earthworm. And earthworm's just going to be my generic term. We're going to look at night crawlers. All right, so earthworms, several species. So it's going to get consumed, which means then it's going to release the sporozoites. And the sporozoites are going to be in the gut. All right? Now, what's going to have to happen is that these sporozoites have to penetrate out of the gut to get to their final site of infection, which is in the testes of the earthworm. All right? So how does it do that? It's going to penetrate the gut. So this is penetrate and migrate. It's going to penetrate the gut, get into the dorsal blood vessel, migrate to the heart, to the dorsal aorta, burrow out of the dorsal aorta to get into the testes. And I have a diagram of the earthworm to kind of show you. If you had zoology, I believe you necropsy, or you dissected a worm, a large worm, right? So you're going to see a major difference when we do it, because our worms are going to be, they're alive. You're going to kill them when you dissect it. So all of the color is there. You're going to be able to see the dorsal blood vessel, you're going to be able to see these seminal vesicles. So your dorsal aortas, the heart part, are up here right near the head, around segments, I think 5 to 7, I think is where it starts. And then the blood vessels run dorsal and ventrally. It's going to take the dorsal and get to the heart, burrow out, and then you are right at the testes and the seminal vessels. I think technically it's called the seminal vesicles because that's where the sperm-forming cells arise. So we're going to say we are the sporozoites now in the seminal vesicle. In the seminal vesicle. Once we get up there, what we will do is the sporozoite will penetrate and transform. All right, so one, what does it transform into? It transforms into a trophozoite. And the trophozoite is a feeding stage. All right, generically feeding stage, it's going to absorb nutrients, it's going to enlarge. All right, so a trophozoite. What does it penetrate into? It's going to penetrate into the sperm-forming cells. It's these cells that produce sperm. And the way you get one cell, and then as it starts producing these flagellated sperm, all right, the cell kind of shrinks until it looks like a furry ball. As we dissect these things, you're going to see these morula, and I have an image here. It's like that could be. This is a sperm-forming cell. These are all the sperm, flagellated sperm. So they penetrate these cells, transform into the feeding stage. And basically, they're feeding on the developing sperm, all right, developing cytoplasm. All right. These trophozoites basically absorb all of the nutrients, all right, until it's basically gone, leaving our trophozoite in the lumen now of the seminal vesicle. So it was like intracellular, but it basically consumed its cell that it was in, and now we have actually a fairly large trophozoite. This trophozoite will then transform into one of two stages. They're transforming into a gamete, is what they're transforming into. But we can't tell much about those gametes. What we do know is that the gametes, each of these gametes are a different mating type. All right, so we've got like a mating, I just said like a mating type 1, mating type 2, kind of like male versus female. So mating type 1, mating type 2, just again, that's just making it out. And this is, because we can't visually distinguish between them, this situation is called anisogametes. The gametes are indistinguishable from each other, but we've transformed into our gametes, these gametes. And then these things are going to attach they're going to attach to the epithelium of the seminal vesicle. All right. Once they attach to the epithelium of the seminal vesicle, now these two gametes of the two different mating types will fuse. In a process called syzygy. I don't know how many Y's are on Scrabble, but this is a word that you go for. Syzygy. You have a Z in there too. Big points. All right. What is syzygy? It's just simply the pairing of the gametes. So we're pairing up. We're pairing them up, and we're getting them together. So now we have paired gametes. All right, and our paired gametes now are going to be like this. So let's say that is our epithelium. You've got these trophozoites that transformed into gametes. The gametes then get together, they pair up, that's syzygy, and then we will fuse and lay down a cyst wall. So this, the membranes themselves will fuse. So fusion, fuse, and cyst wall production, that now gives us a gametocyst. I shouldn't have said use the word gametes. Those are gametes. So we have a gametocyst with two gametes. So now it's the same thing. We've got this structure here where we've laid down the cyst wall, and our gametes now look something like that. So we've got an image where what one looks like. And these are fairly large. All right? We're going to see these you're going to see them at 4x and at 10x. No problem. All right, so once we have this, we went from haploid, haploid, haploid. We paired. Once we fused, we now are diploid. All right? So now, well, no, not yet. Not yet. Well, yeah, not yet. We're still haploid because each of our gametes. We haven't produced our gametes yet. This is our next step, gametogamy. Gametogamy is the production of gametes. So our gamete undergoes gametogamy to produce gametes. So we've got now a gametocyst. Gametes, we don't have micro or macro gametes. Right? They're indistinguishable. And isogametes. Now, inside our gametocyst, we will have fusion of the gametes. And we will have secretion of a cyst wall. All right, so we hit now our 2n number. We hit diploid. Now we secreted our cyst wall, so now we have a gametocyst with oocysts. Running out of room. And then, all right, so we got gametocyst with the oocyst, so now you're dealing with something like this. You've got all these oocysts inside the gametocyst. All right. Now, what we have is a process called sporogony that's happening. Now, the terminology is kind of iffy because sporogony is the production of sporozoites. Right? This is when we're going to see our zygotic meiosis, the reduction. So what I'm going to use is sporulation. Because typically sporulation in other organisms can involve meiosis. Sporulation is the reduction of our gametes, to produce a spore. So we're going to have sporulation, and that's going to be followed by sporogony. All right. Sporocyst and sporogony. So that will be our division stage. And this is occurring within each of our oocysts. All right, so now, once we have that, we have our gametocysts with our oocysts with sporozoites. How many sporozoites? 8. So first division, first meiosis, gets this reduction. Right? And then we're going to have rounded division to get our sporogony, to get to 8 sporozoids. All right, so then the 8 sporozoids, this is where things kind of get a little muddy. This isn't muddy enough. All right, so the gametocysts can get released, or they can rupture and the oocysts get released. All right, so what we're going to say is this. Released or rupture and release. Rupture or rupture and release. Now, where do they get released from? They get released from the genital core of our worm. I think it's probably less important. Because we're in the seminal vesicle. All right, so there it is. Our host is the earthworm. We're outside. Our earthworm, the next host, can either consume one of these mature spores or mature oocysts, aka oocyst, or they can consume one of these gametocysts. So it exits, and then a worm consumes one of these gametocysts that have the oocyst. Either one can. Our mature spores are going to be football-shaped. Got a diagram there. Very easy to tell. Very easy to identify. Adaptations to increase transmission. I think there's probably only one, and it's this spot here. These can survive the GI tract of birds and mammals. So you can have birds consume an earthworm that's infected. All right, if that earthworm has these gametocysts in them, it can pass through the gut of the bird, and then when the bird's flying around and defecating, it's spreading these oocysts out in the environment. That's, you're gonna see, I don't know what the prevalence is if we go out and like survey the earthworms. I don't know what the prevalence is, but it's supposed to be fairly easy to find. As I said, in these nightcrawler farms, I mean, it's every nightcrawler that I've examined has had it. So there's a lot of information there. Any questions? So on Monday in lab and on Wednesday in lab, we'll dissect an earthworm. We'll look for these. Our goal is to find the different life cycle stages. So to find, we can typically find a trophozoite. It's going to be hard to identify trophozoites and gametes that are together. All right, but we're going to be able to see the gametocyst that has these gametes in them. We're going to see different stages of this. We're definitely going to see these gametocysts with the oocysts, and we're going to see the oocysts of the spores, no problem. All right. All right, 11:50. Y'all have a good weekend. And what we will do on Monday is talk about the other Gregorin parasite, or septate Gregorin. And we'll go through the life cycle, and that means then next week in lab we can kind of review all of the organisms that we have in preparation for our practical, which is in a little more than a week.
Is it okay if I leave a bit early in class on Monday to go to
No, you have to stay here till 11:50. Yeah, no problem. No problem. You guys are presenting. So if you needed to, we're live streaming, it'll be recorded. And then if you run late to lab, I know what's going on. Our grad class, theirs is at
4, I think.
5:15. Then they do like the grad student award ceremony afterwards. So we're not even going to do class because we have a couple presenting, and I said go. The grad students, I think they should go. So.